Today, I'm Yuan Zhang from Institute of Information Science at Sinica. The team leader of our group is our Zhongyuan Lin and Dr. Jianming Hou. Yeah. Our research topic is an integrated solution of genome de novo assembly for precision medicine and genome breeding. Motivations. Existing genome assembler often designed for single-order sequencing technology and inherit its inherent deficiency. For example, the next-generation sequencing short read is accurate but too short. The third-generation sequencing long read is long but with about 10% error rate. So a new technology, standard genomic link reads has low error rate as NGS, but also provide long-range DNA information. Okay. However, the advantage of link read needs new algorithm to unleash its full power. Our goal is to achieve more complete, personalized, and non-model space genome while combining the advantage of each platform and assembling with high efficiency. Our method is named GABOLA, de novo genome assembly based on localization and local assembly of scaffold for precision health and genome breeding. The input data includes genetic genomics link reads, Illumina short reads, and long read, for example, the NeoPol and PacBio. And there are four modules, main modules of the algorithm. Local context-based filling, local context-based scaffolding, global context-based filling, global context-based scaffolding. Okay, scaffolding with link reads. Okay, the idea is as follows. Okay, here we can see this is a scaffold, and we map the link read to the scaffold, the draft assembly. Okay, so the link read is different because it has the barcode information for the long-range DNA information. Okay, so those reads belong to barcode 1, those reads belong to the barcode 2, those reads belong to barcode 3. And the center rectangle, these are missing reads. So we can use, we can collect all the reads of barcode 1, barcode 2, and barcode 3 and do the local assembly. And then we will find the candidate context, and if it can align well to both ranking of the gap, we can use it to fill the gap. Similarly, we can do a local context-based gap filling. This is two scaffolds, scaffold A, scaffold B, from the tail of scaffold A and the head of scaffold B. We can also do the local mapping, remapping, local assembly, and try to fill the gap between the two scaffolds. Okay. The two modules are gapping with long read and context. And you can use long reads or context from whole genome assembly. I think this part is more straightforward. You can use context map if it can align well to both ranking. We can use to fill the gap. The scaffold part is similarly. Okay. Our method support multiple scenarios and iterated genome assembly. If you have only 10x data, you have 10x genomics and bionano, you have NeoPol and PacBio, or we provide multiple scenario pipelines, to solve those problems. Okay, the experimental result. Okay, GABOLA versus supernova. Supernova is the 10th genomics official genome assembler, widely used in personalized and non-model genome assemblies. Yeah, compared to supernova assembly, GABOLA assembly provide more genetic found and more genetic information. Okay, for example, 8,500 exome, and so on. More transposon, more region related to gene regulation. And also we have better resolution for repeats. Okay, so for sign, we gain the gain of GABOLA is 2%. Okay, the 9 is about 2.7%, and so on. Okay. This is the assembly statistics. You can see supernova here and GABOLA here. This is the gain of supernova. And we use less scaffold, and the N50 size increase about 31%. Okay. N90 increase much more. Okay. And totally, the gain of DNA size is about 23 mega base pair. Okay, GABOLA versus supernova, or non-model species. Okay, for the Japanese eel, the supernova, the assembly generate, the result is about 500 scaffold. This is 131 scaffolds. Using GABOLA and high-C and population approach, the scaffold number reduced to 144 and 128. And also the boost code, the gene content estimation is good. 94.6 and 98.6%. And with the marker fit, using the marker mapping, we can map all the scaffold to the real chromosome numbers. Okay. Okay. For non-model species, with our assembly, the complete assembly, complete genome with AI for genome breeding, First, you can, for example, you can discover the phenotype specific single nucleotide variation and map to Taiwan telepathy genome to get the VCF files. Okay, and we can identify the phenotype specific markers and do a prediction in AI. For example, the anti-disease fast growth. This is the application of GABOLA. Also, we have a microarray related to this research already created by the company. Our collaborators. Collaborator, include the IBMS for the exhibiting car, or the ICOB at the Masinica, and okay, and also we can the collaboration applications. These are collaborators. Thank you for your attention.